Hello everyone, myself Om Prakash Katte, and today we will discuss the principles of inheritance and variation chapter of 12th class. And topic we are going to discuss, it is inheritance of two genes. Students, already we have learned about the inheritance of one genes. In monohybrid class, the ratio 3:1, phenotypic ratio, and genotypic ratio, it was 1:2:1. Another now inheritance of the two genes. So in inheritance of the two genes, first we should understand dihybrid class. What do we mean by dihybrid class? Because dihybrid class related many questions comes in the examination. So dihybrid class is a class between two pairs of contrasting characters. If two pairs of contrasting characters are crossed together, that class is called the dihybrid class. It means in this class there are the expression of the two genes. Now, so it is called inheritance of the two genes. And many times question come, what is dihybrid class? It is a class between two pairs of contrasting traits. Students, I have taken one example in a pea plant. In pea plant, first one, yellow seed is a one characteristic. So yellow seeds is a dominant characteristic. Yellow seed color is a dominant characteristic, and green seed is a recessive characteristic or recessive trait. So one characteristics I have taken, it is yellow seed which is dominant trait over the green seed, and other I have taken round seed. It is also the dominant character over the wrinkle seed. So it means there are two pair of contrasting character. One pair yellow seed and green seed, and other pair is round seed and wrinkle seed. As I told you, yellow seed is a dominant character over the green seed color. Round seed is dominant characters over the wrinkle seed. Now students, yellow seed, for yellow seed I am taking the genotype capital Y, capital Y. Whereas for the green seed I am taking genotype small y, small y. Similarly, round seed is the dominant character, genotype I have mentioned, I am taking capital R, and wrinkle seed genotype is small r and smaller. So this is phenotypic character and this is genotype. While the formation of the genotypes, when genotypes are formed, now from this homozygous parent and from this also homozygous parent, we have to, they are producing the gamete. So it is homozygous parent, so it produces the gamete capital R and small r. This recessive parent produces the gamete small y and small r. Now gamete of this parent and gamete of this recessive parent, both they are crossed. So in F1 generation, in first generation, we are getting the progeny, and this progeny is having yellow seed and round seed, because yellow seed is a dominant character over the green seed and round seed is the dominant character over the wrinkle seed. So in F1 generation, yellow seeds and round seed plants are produced. So all plants will be having the yellow seeds and round seeds, and genotype of this F1 progeny will be capital Y, small y, capital R and small r. So it means these gamete get mixed up. So they are coming in the F1 generation. So F1 generation progeny is having yellow seed and round seed. Now second, this F1 generation progeny we have to self-pollinate. We have to cross. So here is a self-pollination of F1 progeny. So if F1 progeny gets self-pollinated, in such cases, we have to make the gametes of the F1 progeny. So while the making of the gamete of F1 progeny, look at here, the F1 progeny I am writing separately, capital Y, then small y, capital R and small r. So what type of, how many gametes will be formed? So first one, capital Y, capital R. First gamete will form capital Y, capital R. Then this capital Y can cross with the small r. So second will be capital R and small r. This is the gamete. Third gamete, this small y cross to the capital R. So when small y is crossed with the capital R, but capital is dominant character, so I am writing capital R and small y. And last one, this will be this. So it means small y and small r. So from this F1 hybrid, we are getting the 4 gametes. As I have mentioned, capital Y, capital R, first gamete. Second, capital Y and small r, second gamete. Then capital R, small y, another. And then small y and small r. So these are the gametes of the F1 progeny. Now one F1 progeny of male is having this same gametes. So capital R, small r, capital R, small y and small y. Similarly, I am taking the another progeny which is having the parent, that is a female. So one male gamete and the same gamete for the female also, because it is the self-pollination. So once the gametes are this one male gamete and these are the female gamete. So once now you will cross this capital Y, this male gamete and female gamete. This is female gamete cross with male gamete. So capital Y, then capital R, capital. This cross with this first and go through this one column. Now capital Y, capital R cross with capital Y, small r, then you are getting capital Y, capital R and small r. Then here this cross with the this. So capital Y, small y, capital R and capital R. Now y are this one cross with this one will become capital Y, small y and capital R, small r. So now one gamete of the female cross with the all male gamete. Now second also we have to do. Second gamete is capital Y and small r cross with this one. So capital Y, capital I and capital R, small r. Second, then you will cross this capital Y, small r, and thus it will form capital Y, capital R, small r. Then this capital Y, small y, then small r. And now third we have to cross with this one. Then this one and this one. And last one, capital small y and small r gamete is crossed with the this one. So what will come? First this small y, small r cross with this first table, capital Y, capital R, small r. Second, capital Y, small r, small r. Next, capital Y, small r, small y and small y. And last is small y, capital small r, small r. So thus you are getting the 16 progeny. Thus we are getting the 16 progeny, and these progenies of the F2 generation progeny. So F2 generation we are getting 16 progeny. So dihybrid cross, total number of progeny we are getting, that is the 16 progeny. Now in this 60 progeny we have to go for phenotype ratio. So it is capital Y and capital Y. So capital Y and capital Y is allosite. Capital R is round seed. So remember student, if capital Y come, that will be allosite. If only R will come, that will be round seed. Either they are 2 in number or they are 1 in number. So either one allele come, capital Y allele come, then allosite will be. If capital R allele come, that is round seed. Okay, so if this will be absent, in such cases, their recessive allele will expect. So what to understand now, we have to go for the phenotypic ratio of the dihybrid cross. In phenotypic ratio, look at it. First one, capital Y and capital. Capital Y is allosite and R is round seed. So this is the allo and round seed. Then we should go allo and round seed, yellow 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 and round seed. So thus how many we are getting? 1, 2, 3, 4, 5, 6, 7, 8 and 9. 9 progeny or 9 plants are having yellow seed and round seed. Total 9. Now once which you have counted, you leave it now. So second part is now this one counted, this one. Capital Y, capital Y. It means yellow seed. Small r, small r. Small r is for wrinkle seed. So yellow seed with and wrinkle seed. Yellow seed and wrinkle seed. It should be capital Y1 and small r also. So here capital Y, capital Y, capital Y, capital Y. This is capital Y, capital Y. It means yellow seed, small r, wrinkle seed. So wrinkle, yellow and wrinkle, this one. Yellow and wrinkle, this 2. This is the yellow and wrinkle 1. Then this is the yellow and wrinkle 2. Then already these are counted. Then another is yellow and wrinkle. This is yellow and wrinkle. So thus we are getting yellow seed and wrinkle seed. Total plants we are getting 3. Then another is this one counted, this counted, this is also, this done. Now this is RR. RR is for round. And Y is for green. Round and green. Round and green. So to see round and green, round and green and round and green. So 1, 2, 3. 3 plants are having the round seed and green seed. This is the 3. Now after overall 1 is left. This is this one. And in this case small y, green seed, small r. This is wrinkle seed. So green seed and wrinkle seed. Here you will see green seed and wrinkle seed. The progeny is 1. So overall what happened in dihybrid grass, if F1 progeny is self-pollinated, then we are getting the 16 progeny. In this 16 progeny, the ratio comes. That ratio is phenotypic ratio in dihybrid grass. It is 9:9 ratio, 3:1. So phenotypic ratio in dihybrid grass, many times question is asked to you, what is the phenotypic ratio in the dihybrid grass? So we have to write the 9:3:3:1. As you are knowing, monohybrid grass ratio, phenotypic ratio is a 3:1 in monohybrid grass. Now dihybrid grass ratio, as I told you, 9:3:3:1. Now 9 is yellow seed. So this one 9 is having the yellow seed. Then 3 is yellow seed with wrinkle seed. So it is the 3 is also yellow seed. So look at it. Yellow seed 9. Yellow seed 9. So this ratio again comes 3:1. If you will see monohybrid grass, the ratio is also 3:1, and same conclusion is brought by the Mender in dihybrid grass also. Here this yellow seed and yellow seeds appear. This ratio is also coming the 3:1. If you will see green seed and round seed and green seed and round seed. So green, this is also green, round, green are 3 and green seed and wrinkle 1. So this ratio also comes 3:1. It means either monohybrid grass or dihybrid grass, in both the cases the ratio comes 3:1 if you are going the phenotype. But you must remember phenotypic ratio in monohybrid grass 3:1 and dihybrid grass the ratio is 9:3:3:1. This one by the chart. And this type of any genotype you can take. So if it is dominant character, capital letter you should write. If it is recessive trait, then you should write the small letter. Now another part, this is required for the only 12th class. One more thing we should remember here, if you will see genotype, so capital Y and small r, it is a 1 in number. Such type, capital Y, small r, capital R and small r is 2 in number. So thus, if you will go for the genotype of the 16 progeny, so here ratio come 4 times 1. 1 is to 1 ratio 1. 4 times 1. 4 times 2. 2 ratio 2. And 1 time is come the 4. So in the hybrid class, the genotype ratio, this I have written the genotype. So all the genotypes of this combination and the ratio come 1 ratio 4 ratio 2. But you should remember only the ratio of phenotypic ratio, and that is dihybrid class. Students, one more thing I have to tell to you in the dihybrid class. Here capital Y and capital R. It is dominant homozygous parent. So if you prepare the gametes of this one, so all gamete will be capital Y, capital R, capital Y, capital R, capital Y, capital R. If homozygous. If homozygous recessive is there, suppose small y, and this is small r. So in this case, alleles will form small y, small r, small y, small r, small y, small r, and small y, small r. So it is occurs, or I have written capital R, capital Y, capital R, all these progeny, and here it is small y, small r. But if heterozygous parent is there, so this parent is heterozygous. So in such cases, heterozygous parent, this type of gamete will be formed. As I told you how to take out the gamete of the F1 progeny. So it is gamete formation in homozygous parent, and if heterozygous parent, again I am revising this one, capital Y, small y, capital R, small r. So gamete will form capital Y, capital R, capital Y and small r, then capital R, small y, and last small y, small r. So overall is what? It is the expression of the two genes in this two pair of contrasting character we are classing. So yellow seed is dominant over green seed, and round seed is dominant over the wrinkle seed. So F1 progeny we are getting yellow seed and round seed. But if gamete are forms from the F1 progeny, I have taken male gamete and this female gamete, and when we are crossing this, that 16 progeny we are getting, and ratio is yellow seed and round seed 9, yellow seed and wrinkle seed 3, green seed and round seed 3, green seed and wrinkle seed 1. So thus overall we can see in dihybrid class the phenotype ratio comes 9 ratio 3 ratio 3 ratio